Hello everyone, I'm here to present the results of my work, Dominance Hierarchy, Fertility and Cuticular Hydrocarbons in the Primitive Eusocial Wasp, Miscocyterus cerberus. This work was performed by me, Rafael Carvalho da Silva, and my fellow collaborators. Primitive Eusocial Hymenoptera are represented by several species of ants, bees, and wasps. They are classified like that because there is an absence of morphologically specialized cases among the females, and basically all or at least the most part of the females is potentially reproductive. Females in these societies use different sources of communication which help them to regulate their social environment, such as physical, visual, and chemical sources. Alpha females or queens in these societies usually use different strategies to keep the cohesion in their colonies. In this context, non-reproductive females might benefit by recognizing and avoiding conflicts with them. At the end, it means less cost for either queens and all the other females. The social dynamics aspects of several primitive social species are already known, such as for Gymnoponera quadriceps and Polychus satin. However, this is not the case for Miscocyterus wasps. Considering that Miscocyterus share a common ancestor with Polychus paper wasps, it is expected that they both share also common strategies to keep their colonies functioning cohesively. Being that said, our hypothesis was that females or queens use aggressive and chemical strategies to maintain their dominance in nests of Miscocyterus cerberus. For that, nests in two different phases were used, in pre-worker and post-worker emergence phase. In summary, we basically investigated the link between the aggressive acts performed by females in their nests with their reproductive status and also chemical expression. Additionally, we also performed some nest manipulation to check whether reproductive and chemical traits change according to their social position. All the study was performed in Ribeirão Preto, in São Paulo University area. The behavioral phase consisted of videotaping females to understand their social interactions. For the experimental nests, after videotaping females, the most aggressive one was removed and a new set of videos were made to check the changes in the social dynamic. After collecting behavioral data, all females were collected and taken to the lab where they were used in chemical and physiological analysis. For all videotaped nests from both studied phases, it was possible to spot always a female that performed the most part of the aggressive acts toward the other ones, and these females were classified as alpha. The females that received the most part of the aggressive acts displayed by alpha were classified as beta females, and all the other females were classified as subordinate ones. Females did not differ in terms of body size, however, alpha females, which were the most aggressive ones, also had the biggest oocytes, indicating a higher reproductive status if compared with the other ones. All the alpha females were inseminated, but a high rate of insemination was also found among the other females. As we can see here in the PCA graphs, females in both studied phases are chemically similar to each other. The variation detected, however, was small and restricted to the presence of one or a few compounds in alpha females, or at least the upregulation of a few CHCs. Concerning the removal experiments, after videotaping nests and removing the original alpha, we saw that in the most part of these experimental nests, the female who replaced the original alpha was previously a beta. The new queens were similar in terms of reproductive and chemical traits when compared with the removed ones, and the most part of them were also inseminated, indicating their capacity to take over the reproduction in the nest. Although all females are similar in terms of body size, our data confirmed that some traits differ among them. However, these differences were not as evident as it's possible to detect for some police societies. The fact that a single female is usually the most attacked is probably linked with these females being the most likely to take over a queen position and may probably represent a threat for them. So, alpha females probably use aggressive acts to suppress these females. It's also possible to think that the alpha characteristic compounds found in both studied phases are probably involved in a kind of communication among alpha and the other females. However, it's worth mentioning that in Polycis-Satan societies, the synthetic versions of the queen characteristic compounds were not able to suppress worker reproduction when applied over the nest surface. Instead, workers were seen to be suppressed only in control nests, in which the queen was present. Thus, all this evidence suggests that in these primitively social species, these alpha or queen characteristic compounds, although found in a higher proportion over the cuticular surface of alpha females, they merely represent chemical cues that are produced as a consequence of overactivation or aggressive levels, but do not act as signals. In summary, we believe that in societies of Miscocyterus cerberus, alpha females may use their aggression as the main strategy to control all the other females, despite of the nest phase. The small chemical differences found among females from different groups may be used by the other females to avoid unnecessary conflicts with the alpha ones, especially whether these chemical cues are linked with their reproductive or aggressive status. The presence of chemical cues related to fertility might be useful for females in post-worker emergence nests, especially if a single female reproduces and the other invests their energy in expensive tasks. These cues could contribute for keeping the cooperation among females. So that's all. I would like to thank you for listening to my presentation, and I will be glad to answer any questions if you have. Feel free to contact me.